So I'll take a moment. There'll be, like I said, this is a handout as well, and so you can always go back, but I'll take the time to stop, and I promise we'll at least make it so that you can do basic spectral interpretation, and most importantly, watch out for the many roadblocks that you're going to run into when you're actually a metalloenzyme person trying mass spec. Okay, so why would you even do mass spec? I was at this same conference in 1996, 98, 2000, teaching MCD, EPR, resonance Raman. They're way cooler if you think about, the nature of the techniques. They're quantum mechanical. You got to love them. But I had let myself do whatever I wanted as a PhD student. I said, I'm just going to go learn and have fun and see what it's like to be a kid at Oxford or University of Georgia, as it were. But when I got out of, when I left that, I really wanted to study a disease. And it occurred to me about 2 years into my PhD, MCD, Raman, EPR, I need milligram quantities of proteins, and I want to actually purify something from a person and find out what's wrong with it. So necessity made me go into mass spectrometry, where if you put more than a picomole, which for most of us is some number of nanograms, some low number of nanograms, if you put more than that on our instrument, we'll be mad at you. And if you give us an animal, we have a very good chance of detecting it. So don't underestimate the 6 or 7 orders of sensitivity, orders of magnitude of sensitivity, what they can bring you. Even if you're not going to go into the field, all of you are making proteins one way or the other, or using somebody else's proteins. And I can tell you from experience, and it's terrible, only about 1 out of every 7 or 8 is our best estimate now of seeing 70, 80 proteins. Only less than 10%, or that was more than 10%, let's say 10%, are not what you think they are. They don't even have the right primary sequence. Does anyone want to guess where the problems usually lie? N-terminus. You never really know what's on your N-terminus. You put a his tag, you imagined you'd cleave it. You didn't really think about how your codon usage optimization would work out. You didn't really think about your restriction fragments, and you have the wrong N-terminus. But often it's other things. We've had people give us disease-causing variants, and that's what they've been studying for 5 or 6 years without knowing that they were inadvertently studying something that causes a disease in their very same protein. So you got to do the quality control. Something that's really underestimated, we are all used to thinking about this sort of 10 angstrom or even 5 angstrom shell around our metals, right? That's the cool place. But there are other in vivo. The truth is that other post-translational modifications, be they structural, be they things like phosphorylation, cysteinylation, they modulate the whether you even have a metal. A disulfide is a great example. Very many metalloproteins require a disulfide either in their metallochaperone or in themselves. Some sites of metallation are blocked, so you can go in vivo and look for these. And later in this talk, Jared Auclair will tell you a few examples about that. Again, this is how you get outside of your 10 angstrom bubble. Get outside of it with mass spec, X-ray crystallography, NMR. Again, compared to X-ray crystallography and NMR, which we complement, I'm not trying to compete with them, we have a major sensitivity advantage. You don't have to crystallize the protein, and you don't have to solve a structure. So it can be very fast, which I'll get to in the end. Hopefully by the end of today's talk, I will convince you that you can also study reaction mechanisms. We've done this. If you look back into the metallo, like ISCU, I did this as a PhD student. You could watch sulfur add. You could watch iron add. You can even watch a single electron add, either by its relativistic mass or by the way it changes the charge state distribution. I said this already. There's a growing field where mass spectrometry can actually provide the constraints for molecular mechanics and de novo structure assembly of a protein. The MM people are getting closer and closer to be able to solve something really de novo. And in some cases they can, rare cases. Again, it's fast. We can do an analysis in a minute if we have to. Okay? So that's one reason. And I'm going to just get out here and say it and challenge you to tell me otherwise. By far, we have the most sophisticated instrumentation. You can't touch us. Now, I say that having done many other things. The only thing that comes close is a synchrotron. And why is that? I'm going to back it up really quickly. Things like this, things I'm not proud of, but that's the Manhattan Project. And if I can figure out how to do this laser, you said this, Jared? We may have the most sophisticated instrumentation, but not the most sophisticated users. That is a person, that is a single mass spectrometer, more than $1 billion, not of today's money, of those days' money, were put in. That's one of those mass spectrometers you just looked at. They made tracks of these and buildings of tracks. And notice the first operators of mass spectrometers, and they were all women, by the way. You can wonder why that might be. And there's Lawrence, Nobel Prize winner for the cyclotron, the instrument that much of what I'll talk to you about is based upon. Here's another reason. Every single spaceship that goes up, and indeed even astronauts, have a very small mass spectrometer or a larger mass spectrometer to monitor things. And finally, when you're in the airport and they're testing you, those are mass spectrometers. So it just turns out that there's been a lot of, I'm not like, ooh, we're better, but it's just a matter of truth that there's a lot of research dollars that went into developing these. So it's the simplest, it's strictly Newtonian for the most part, and yet it has the most sophisticated instrumentation. And what I can't tell you about today is how to use that to do all the fancy kinds of things that involve breaking proteins. What I want to just get you to is the first phase of measuring the mass of your protein and saying, oh, it has a metal, it has the right metal, it's the right protein. How does the metal affect the structure? Once you have that data, you can convince any mass spectrometrist to collaborate with you because you will be one of the handful of people in the world, all of them trained through these channels, who can actually get a spectrum of a metalloprotein. So, this is Moore's law. We've all seen it. It's the number of microprocessors you can squeeze into a chip, right? Cool. And it actually goes back even to electromechanical stuff, which I find neat. And some people argue to the abacus. I don't. The genomicists get up there and they say, look, this is Moore's law. Look what we've done. And they can take pride in this, and we've all been part of it. I was there and got the first science paper when the first genome was solved, and that was just amazing. So what about mass spec technology? How's it doing? By the way, if you have a log linear, this is exponential explosion right here. What are we doing? Well, I plotted this myself, and these are the number of proteins that you can identify by mass spec, which in 1983 was one. Really. There were a couple Nobel Prize-winning innovations, electrospray ionization, you probably all heard of it, ESI, and MALDI. Both of those won their inventors Nobel Prizes. And then, once that happened, the mass spec technology was already there by and large. It just needed to be speeded up because it had been built for years, right? And here we go. And in fact, it's getting a little bit better here. And finally, we have a comprehensive proteome. It's now the case that we can measure all, in quotation marks, of the proteins that are in a cell in terms of identifying them. We do not have 100% sequence coverage. Sorry about that. I'll try not to do that anymore. Okay. And to give that, put it in perspective, 10,000 phosphorylation sites and all this. Now, who cares about phosphorylation except that I'm telling you the story of my life has been that one post-translational modification affects another. And if you're ahead of the curve on that, you'll do well or do better. So why learn mass spec? Well, this is the bad news. You have to be the expert if you're a metalloenzyme person. Your facility, this is true for 90% of the facilities, are made to do what? Proteomics. And what do proteomics people do? They chop up proteins into little pieces, your metals leave, and that's the end of the story for you. But it's worse than that. You won't get data because they don't know how to prepare the sample. I'm going to teach you that. You're going to break your protein, and you're going to lose the metals. We just said that. You're probably also going to break the mass spectrometer. And that's no joke. When I started studying superoxide dismutase, I brought it to the facility, and I had to have $1,000 in my hand just to get the columns replaced because I broke it every single time, the column. That's still true. And if you get signal, this is really important. You need to be paying attention at every level of this. Because even if you get signal and you have a protein mass spectrum, guess what? The mass is going to be wrong that your facility gives you back. 95% of the time or more. So I'm just being nice so you can all think that your facility is the one that doesn't do it incorrectly. But so it's not their fault. You would do the same thing, and even the canned data analysis programs are going to do the same thing. They are going to all be based upon the assumption that during ionization, protons that weren't there in solution went onto your protein, protonated it, gave it its reason, and made it able to go into the mass spectrometer, because mass spectrometers only analyze ions. And they're going to assume that every charge came from those protons. And so once you're done measuring the mass in the mass spectrometer, what do you have to do? Subtract those protons. Guess what? If they're holes, you're in a lot of trouble. You better not be studying molybdenum-6 or iron-4 or copper-2, because every mass is going to be off by the number of holes you have. And that's significant. So we've said this, but it bears repeating. You just got to hang in there for another hour. Okay. So before we get into the nitty-gritty of mass spectral interpretation, I'm going to give you a series of things that, in fact, most people don't publish. And there are ways to get a signal from a metalloprotein. So what do you need to know, first of all? When somebody tells you they have a pure protein, right, it's not really pure in the mass spec sense. It has salts, buffers, detergents. Personally, I spent 6 months where I could get a MALDI signal on something that I was purifying from human brains, and I could not get an electrospray signal. 6 months of a postdoctoral career were lost to that single issue. And it turned out to be detergents, not any that I put in there, I knew better, but that just permeate molecular biology labs. So I had to set up a detergent-free zone and guard it with ninja moves until I could get it, and that worked. Also learned at IBSW 2000, the ninja moves. Okay, so you got to get rid of the crap. This is it. Take your sample, and if you can do this, concentrate it in a Centricon, put in some ammonium bicarbonate, put that through a few times, then put in some, if your protein will survive, just HPLC grade water. All your water has to be HPLC grade. If your protein won't survive, put in ammonium acetate. Okay? And this is often all you'll need to do mass spec. It's pretty simple. But that's only if you didn't put detergents in there. If you put detergents in, don't ask me what to do, because I just avoid that. Okay. We all know this, but I'm just going to say this. Proteins are not small molecules. You guys know how to treat proteins, except we won't say any names, but one of the illustrious people who study ribonucleotide reductases sent a sample to one of the illustrious people who study such proteins by magnetic circular dichroism, and that'll narrow it down, but it won't let you solve it, and said graduate student left the protein on the bench. Just for a few hours, some say overnight, no one really knows, the protein died. The bad news is that took a year to purify. So said magnetic circular dichroism student had to go back to said FNR lab and spend 6 months purifying their own sample, because said other scientist is really someone you don't want to mess with. So don't leave your protein on the bench. Freeze it quickly in nitrogen. You should know this, but I have to say it. Because mass spec we see things that you don't. Again, if by you're somebody who uses EPR, MCD, XAS, anything, even crystallography, what you're not going to see is all the broken pieces of your protein. And when you send it to us, we see them. And so I tell you, all the freeze thaws that you think aren't hurting, when we get the protein, I've had people give us things, and I would ask, is this a protease digest? And they would say no. And they were NMR labs of note, and had published that data. And that happens a lot. So again, remember, we're getting outside of your comfort zone, but this is good. Okay. Large proteins, this is still something that is unsolved. Just like with crystallography. If you can get a crystal structure of cytochrome C oxidase, you deserve a Nobel Prize. If you can do a membrane protein, good for you. And there aren't great techniques still for that. So I want to put that out there and say, that remains to be solved. Okay, so detergents are bad for electrospray ionization. Keep them out. There are some new ones you can spend a lot of money for. Just remember sugar-based. Those will help you. You cannot get rid of detergents like you think you can. It's just like, of course, all they have to do is dilute them to below the critical micellar concentration and then centrifuge. It doesn't work. And filter, that doesn't work. Okay. Reverse phase also won't remove detergents, not well. And they can actually help MALDI. So if you're not getting a signal, you can put detergents in and get a MALDI signal of your large protein. Okay. This, I'm going to really go through quickly but this is, again, I made this here, workflow for protein analysis. If you have detergents, you have to go this route. And you have a 1 in 10 chance of being able to get a metalloprotein spectrum, but there is now, and I provide a supplementary sample prep guide at the end of the lecture, so you can go to it yourself and print it out of how to do this. But what most of you are going to do is hopefully keep out the detergent. You want the intact protein's molecular weight, and you do top-down mass spec. Do what I told you about the ammonium bicarbonate. You might use C4 reverse phase if you keep the acid out of there, you might actually maintain metals, but your facility is going to have acid in all of their solvents. So you have to be wary of this. Formic acid is what is known as an ion pairing agent, and it helps the chromatography, okay, by eliminating charge interactions, but that doesn't really matter. Okay? And then the rest of this, for every single one of these, even if you want to just characterize your protein's primary sequence, any one of these will work, and again, I give you protocols for all of those. Okay. What do we need to know about a mass spectrometer for this? Not a lot. We need to worry about the data, I think. And if you're really interested, you can go read a mass spec book. What you do need to know is that the ionization source. Why do you need to know this? Well, if you're doing electrospray, you put on a variable number of protons to your protein, okay? It could be 1, it could be 10. And so proteins have what is called the charge state distribution by electrospray. Now, that's really cool, because if you think about a distribution of charge states +2, +3, +4, if you reduce your protein by an electron, you've done two things. One, you've changed the mass, because an electron has a rest mass, as does a photon. And we can, by the way, measure photons by mass spectrometry, and I'll show you an example of that. But you will shift your charge state distribution as well, so that you'll have two hints that you've reduced it. One, if you could measure the mass accurately enough to detect the electron, it's more than a hint. The other is if you see your charge state distribution moving. Now, for MALDI ionization, it's simpler. There's only one charge, usually. That's changing now, too. Technology is developing rapidly. But for MALDI, you can have a +1. Now, electron ionization is most of the, we're not supposed to say young or old, but most of the more mature mass spec lecturers will still teach you this. And that's what they used to use for small molecule mass spec, and it uses electrons to knock off other electrons to create holes. These are the only mass analyzers. Sectors kind of gone the way of the dinosaur. They're still great things. You're going to probably be using a quadrupole, a time of flight, or an FT, or some combination of those into a hybrid mass spec. For example, in our lab, we'll be using a quadrupole Orbitrap. That's all we're going to say. This is all mass spectrometers do, despite all the technology that goes into them. I think that's a funny irony. They just weigh things, they purify things, and they break things. That's really it. Yeah. Oh, yours weigh? Wow, I got to talk to Marty. He runs a tight ship. So, okay. So a few things real quick that we have to get out of the way. We have to get you a great thing about mass spec too, honestly. And when I take a mathematician or a physicist and we talk about mass spec, it fills in a gap, a really important gap between your macromolecular world and the quantum mechanical world that we're used to. It's really nice. So we're going to start, and like you put me on a scale, first of all, after you go, oh, you don't want to know how much I weigh, but if you did that, I am an average mass, yes? But that's not as meaningful as a monoisotopic mass, and so we have to get down and realize that if we're looking at copper, for example, stop thinking that that's what copper weighs. Please. Okay? It doesn't. There's never been, I hope, if it did, if copper had that molecular mass, you could use it to make the most powerful bomb in history of the universe, because you could just harness that mass, because it's clearly not a proton or a neutron. It must be nuclear binding energy, and then just blow everything up. So that's not, you don't want to do that, and it's good. But what copper really is is two things, essentially. I mean, I'm oversimplifying a bit, but it's 69% 63 copper, 65% or 31% of something that is an M+2. It's 2 units away. That's great. By the way, if you see copper in a mass spectrum, it sticks out like a sore thumb because of its isotope ratio, and that's true for all transition metals. So the first thing to remember and note, all transition metals have a unique isotopic signature. And I'll tell you later, they all have a unique mass defect, not this though. Okay, and so what that means in a spectrum is something like this. If you just put copper alone, 63, 65, and again, that's a telltale sign. Now, this is a very important concept. I absolutely love it. The mass defect. By convention, carbon is 12.000, of course. So that means that the nucleons, the protons and the neutrons in a carbon nucleus, on average weigh 1.0000. That's what that means. When they're bound, in a bound state, not in the continuum. Okay? If you're a chemist, you can be proud of the fact that it used to be 16.00, the physicists, and there just ended up being more chemists. So I have a bunch of old books that are outdated because they were on a 16 scale. Okay, so if you have a nuclei or a nucleus that's more stable than carbon, right? More stable than a carbon nucleus, and I mean in terms of its nuclear binding energy, it's going to weigh more or less. When you look at the slide, it's going to weigh less because the virtue of stability is being lower in energy. And if you're lower in energy, and E equals mc squared, and delta E must equal m delta c squared, then there you go. Okay? So the nominal mass, you can forget a lot of these things. The concepts you can't forget, but the nominal mass is just the number of nucleons. So it's the number here is nominal. This is mass defect. Turns out that we can use these two alone to assign a molecular formula with a good mass spectrometer. As a rule of thumb, think up to 1,000 Daltons. So just from the mass spectrum, we can say, well, there's only some certain combination that can add up to whatever this is and whatever that is. So, for example, 720.084 can only be one thing. It's a buckyball that somebody protonated 12 times. Okay? We've said that. Mass defects have periodicity as well, which is kind of cool. And get down into the transition metals, and they really make a difference. So the only things that have masses, stabilities greater than that of, or less than that of carbon, are in green. Do you see anything common about these? You can make bombs from them. That's really it. I mean, that's really the mass defect that's being harnessed. It's the energy. It's that mass, that tiny little 0.01. When you multiply that by the speed of light and then squared, that turns out to be a heck of a lot of energy. Okay? So, and here, we're in the other end of this. We're down here. You know, this iron is the best metal. Everyone knows that. But if you study other less cool metals, that's cool. But you still have a mass defect. And that's fine. Copper's not bad either. And molybdenum's pretty cool. Does anybody study zinc? Anybody study zinc? I'm sorry. All right. All right. So with that said, there's kind of an allegory of the cave going on in that there's a real underlying truth to how a mass spectrum is built, and you build it in your mind from the bottom up. You say, I have a carbon 12, and that's my spectrum, but wait, 1% of carbon is carbon 13, so I have to put another little peak there, and we're starting to build a spectrum in our minds, and we say, wait, if I have 100 carbons or 1,000 like in a protein, now the probability of having a carbon 12 is quite large, and in fact, I might not even see the protein that is all carbon 12. That's how mass spectra are built. You can build them in your mind like that, but that's the thing to do when you're pondering and you're alone. That's really how to do it, but the mass spectrometer is going to obscure that. So if you use a MALDI-TOF, it's going to look like this in black. This is a protein mass spectrum, and maybe I should start here. A peptide, where the peak that consists of mostly carbon-12, the monoisotopic peak, if you have a good mass spectrometer, you can separate one carbon 1 carbon-12, 2 carbon-12, and so on. But if you have a bad mass spectrometer, it's this. And so what you get is the average mass, or something like an average mass, as opposed to a monoisotopic mass. Proteins, now I'm going to show you what happens on a really good mass spectrometer when you split this peak, because of the actual differences in nuclear binding energies. And even better mass spectrometrists, well, with better mass spectrometers anyway, have separated polarization. If you can take CO and polarize it, okay, polarize it, and see the 3 states, that eigenstates that must come out of that, the masses corresponding to those eigenstates. That can be even that visible photon. So proteins, again, isotopic resolution and not. So just keep in mind that you have to know the resolving power, which is the mass that you're at divided by the delta mass at full width at half height of the mass spectrometer before you can interpret the spectrum. Okay? All right. When you get your data as a result of, look at this, we have all of these isotopes. We got to get rid of those because nobody, you don't care, even I don't care about all of those masses because they really only project back to one thing, your protein and what its chemical formula is. So to get rid of those, you must de-isotope them. And there are just programs that will de-isotope. And if you had multiple charge states, you must deconvolute those, which is interesting because you're putting them together, so you should convolute them really. But anyway, that's how they speak the parlance. So deconvolution, converting from M over Z to Dalton's AMUs, these are all acceptable. Okay, and then here we go. When you go to interpret a mass spectrum, we'll come back to this. I give you a spectrum in a few slides. The only thing you have to remember, and you guys are going to be well suited for this because you deal with more difficult theoretical material than what I'm about to present you with. If I tell you that these 2 peaks, this is not a good, you don't get to see this. And I tell you the spacing between these 2 peaks is 0.1 m over z. What must the charge state be if this is in units of m over z? Yeah, it's a 10 plus. And teachers aren't allowed to answer. Of course, so it has to be a 10 plus. So delta m by z, that's a neutron that's making that difference. And neutrons don't weigh 0.1, so just go, oh yeah, and multiply by 10 to get that spacing right. You've taken it from m by z into charge state, into mass domain. And you've calculated the mass of this as it is in the mass spectrometer. All you need to do after that to interpret it, so, right, this is 0.1, and we say, well, it can't be 0.1, it needs to be 1. I need to multiply it by 10 to get it to 1, so it's a plus 10 charge state. I do, therefore, multiply it by 10. Now I know what the thing weighs in the mass spectrometer, and now I must ask the question, how much of that weight is false, came on during ionization, and how much of that comes from my metals. And that's where the art comes. But if you're studying zinc, you're fine. Because 2 of your charges came from the metal, period. Unless we could weigh what happens in the crystals and synchrotrons and frozen and stuff. Then you've messed it all up. But okay, so that's that. We've talked about this. You can, again, you can, the relative abundances of the m and the m plus 2 and the m plus 1 can be used. We talked about that. Now we'll get into actual proteins. When you spray, this is an actual electrospray. A nanospray spreads out a little bit prettier. 3,000 volts. If it's nanospray, it's 1,000 volts. You can do either one of those. You may want to do nanospray if you're trying to keep your metals in. If you do what I told you about purifying your protein in ambic and keeping the metals in there, this is what a mass spectrum of cytochrome C looks like. Pretty well defined. Mostly a plus 9, a little bit of plus 10, and a little bit of plus 8. Now, if you do what the mass spectrometrist would have you do and put it in a formic acid solution, well, the metals will fall out post-haste, and this will be what your spectrum looks like. First of all, harder to interpret. Second, charge state distribution is shifted. Now, if I reduced cytochrome C, it's almost pedantic, but really think about it, then well, the plus 9 becomes a plus 8. Okay? And the mass shifts by the mass of an electron, which at the intact protein level is still not possible to measure. Okay? So you can also break it if you want. You can put it in, break it in the mass spectrometer. You can put in superchargers. There's all kinds of things you could do. But this is the fundamental thing to remember about protein mass spec is you can get your metals in there. You can throw in some acid yourself. It will help you get a better signal. So you'll see your apoprotein and your metallated protein. There you go. We can stop now. So now we're ready to interpret spectra ourselves. No teachers may be involved. What is this? This is an M by Z. This is called the monoisotopic peak. The average mass is somewhere around here, the peak corresponding to the average mass. What is the monoisotopic mass of this? In the mass spectrometer, the monoisotopic mass of that is 4,000. That's important. So we just said these are point. Wait a minute. 1, 2, 3. No, what? What's that point? 1, 0.2, 0.3, 0.4. Oh yeah, that's missed. Okay, so anyway, I got a little. Yeah. So it is 4,000. Now we have to ask: does it have metal or not? Because if it has metal, 2 of the charges come from the metal, and if it doesn't, if it's just all protonated, then you just subtract 4. So you either subtract 4 or 2, for example, if you had zinc. All right. Here's what can happen. This is what you have to pay attention to in the near future, even now. If you take a really good mass spectrometer, that's your M plus 1, that's your M plus 2, and you blow these up, now you see everything I told you. That's your carbon, the peak corresponding to carbon 12. There's if you have oxygen 18, carbon, sulfur, and by the way, if there were copper, right, this would be right here. Copper has quite a large mass defect, and so copper would be about right here. Okay? And underlying this for every transition metal would be a unique, what are called isotopic fine structure signature. And when we first did this, one group has done this with proteins, the guy who invented the FTMS, Alan Marshall. We just got a mass spectrometer that lets us do it with proteins as well. And when we first did it with peptides, we couldn't find, there was actually nothing that could fit the data, that could even simulate the spectra. So we wrote the code and developed that, and it's available now. So even to simulate these was an issue. It's trivial in terms of what you have to do, the thought experiment, but the computers had a hard time with it. Okay. Again, this is a good example of, and in this case, we're talking about myoglobin, right? So pH 7, pH 4, pH 4, okay, and then here we go, pH 2. So this is unfolding of myoglobin due to loss of, due to acid, but losing metals and unfolding, and when you unfold, you get more protons during ionization. So there's a labeling experiment going on during ionization, right? If you're tightly folded, if I could fold up into a very high surface area to volume ratio, I only have 1 or 2 places to protonate, and if I'm like this, then you can protonate places that weren't there. So that's the labeling experiment that's going on implicitly during ionization. So you're seeing both those. There's going to be, and if you deconvolute these and deisotope them, this peak, the things corresponding to this peak, the molecules, weigh more by 1 iron. Okay? And are also more unfolded. Now we can also add deuterium at this point to any one of these. If we put deuterium, deuterated water, into this, put these into a solution of deuterated water, guess what? And I'll show you examples of this later. Which would exchange more quickly? The unfolded one. And so over time, these are going to gain mass faster. They're going to have type 2 diabetes, and these aren't because they're more folded. Okay. And again, we like diseases. This isn't my data, but hemochromatosis, everybody, HFE, iron transport, and I'm going to use this to illustrate the state of the art in mass spec, this isn't me doing this. The mass spectrometers in the last couple of years, and it's really in the last 6 months, have become commercially available that let you go to these kind of M over Zs. It used to be 4,000 was a good limit. So you could see your monomer, maybe your dimer. Proteins edge up in M over Z as they get larger. That's the way it works. DNA doesn't. You can measure a plasmid, and it'll have an M over Z of 1,000. Okay, no matter what. Or you can measure a single nucleotide. DNA is cool like that. Proteins aren't. But now look at this. You can see, incredibly large, and I'll just, you can read this if you want to, but this is the protein in the iron core of ferritin. If you can do, now again, this is from 7,000 to 14,000. I have to piece this together for multiple publications because no one's done the complete study yet. If you take the 24-mer of ferritin and you want to break it, it will break down into things that you can interpret as monomers, dimers, without metals. And it's incredible. And there's more to be read about this, but let me just say, big stuff is now in the game. You can now look at large proteins. And I remember looking at ferritin. It had a crystal structure was solved really early, so even as a grad student, you could go enjoy that and think, like, oh my God, when are we ever going to be able to do anything with that little rusty mess? And okay, so with that said, keep in mind, and this is just a reference to that, again, that's just the surface, the iron. I mean, transferrin, for example, it's a glycoprotein. It gets covalently modified by drugs people take. It has so many cool modifications that you don't detect by they're not paramagnetic, and and so on. Okay, so here goes. We want the room to get darker, and this goes. Here, I'll show you an example of some of this stuff now applied to a disease. That's your spectral primer. That's what you need to know. You just need to be able to look at a spectrum, assign a charge state, know that you need to know how many charges came from your metals. If you can do that, you're good to go. What do we do? So what have we been up to in our lab, and hopefully we can illustrate some of these points and extra things you can do with the mass spectrometer. Well, what about a disease? We'd like our disease to be the way we're all trained to think. We're all trained to, well, at least in this group, many of us are trained to figure out how the protein works, right? How does it function? What is the metal doing? How does it react? And these things. Guess what? If you have a dominantly inherited disease and you go to chapter 1 of your genetics book, it's going to tell you often dominantly inherited diseases are a gain of function. Not always the case, but still mostly the case. So it might have nothing to do with your metal. You can't take for granted that the metal is the center of it, and then you have to get outside and ask what's happening to the whole protein. Don't worry. I saw some tears in the back row. The metal is important. So it's fine. It's fine. So, okay. What's important for us is that the gain of function is really unknown. So there's some gain of function that causes ALS in the metalloprotein copper zinc superoxide dismutase. Okay? What's this? This is a primer and spectral interpretation. Marty, you can answer this one if you want.
What is that?
Yeah, what is that? Anyone else want to guess? Could it be an IR spectrum? I saw that. I was like, oh my God, fine structure in life and death. So the real important thing is that you have to understand that people were only living to be 25, 1750. And now we're pushing 80, right? And it's getting even worse. So all of a sudden, humankind has a bunch of diseases that they didn't have before. What happened? What did I miss? Okay. Now, of course, the fine structure is cool too, but the big thing is people are living longer, a lot longer. Now, the fine structure means that, wars are bad. And this is why you don't try to invade Russia in land war. People learn every time, or whenever that was. Okay, so this is what the population curve looked like. This is just a little bit of fun. It always looked like this. There was a pyramid. There's an unfortunate thing that happens with children. Over time, that's bad. There's infant mortality. You're seeing that right there. This is a histogram. And it was always a triangle, and that's why Social Security worked. Because it's not a joke. These people were supporting these people, but now these people are supporting these people. And this keeps pushing up because these are still some old people that had different life expectancies that need to die before this can go up faster. I'm doing my part. I'm dying as fast as I can. So those are the population curves and what they mean, and that they're not, and they look like spectra, which is cool. But this is really terrible. I mean, look at this. This is your probability. This is death. I can't believe this. So this is, what good is this without an axis? It's horrible. That's deaths per 1,000. So the probability for a woman having Parkinson at age, this is Alzheimer, excuse me, at age 95 is 0.5. And people say, although most of us reach 105 and you die programmed death by transthyretin familial amyloidosis, but not familial in your case. Your transthyretin falls out of solution, and then you die from that. But if you could continue to live, you'd have a unit probability of Alzheimer somewhere around 110 if you follow these curves. So you will get Alzheimer if you can live past your programmed death from amyloidosis. And Parkinson's, same thing. So those are things we study. So ALS, what's the question we ask? There's 150 mutations in this one protein. That's SOD1, is short for copper zinc superoxide dismutase. Mutations in another protein, DJ1, that cause Parkinson. And how do 150 things all do the same thing? In fact, you ask the simple question, do they all affect activity? No. Half of them are just as active as the regular enzyme. And when spectroscopists look at them, they can't see a lot of differences, so that's tough. And this all matters because if you can find what's wrong, diabetes used to be a death sentence. Okay? Type 1 diabetes was a death sentence, even 30 or 40 years ago. I only know a few people who had type 1 diabetes and had it before insulin replacement that survived it. And it really was a death sentence, but it was fixed by simple replacement therapy. So if all that's happening is your protein is falling out of solution, there's a bunch of things you can do. So we want to determine that first. If it's just a loss of function, you put more in, it's easy. If it's not a loss of function, there's a new class of therapeutics that are being developed that stabilize proteins, so-called pharmacological chaperones, and you would want to take a second approach to that, and Jared will tell you some of that. So here we have to have a hypothesis. So if a bunch of mutations destabilize this protein and cause ALS, we think they all cause similar structural defects. We'll just say that. You have to have a hypothesis, right? And we also want to say that mutations cause only 2% of overall ALS. That's it, mutations in this protein. Keep in mind, if you have the mutation, if you have like an A4V, an alanine to a valine, can you imagine? Alanine to valine, a methyl group on a protein that weighs 16,000, you will get ALS if you live to be 50. It's unit probability, so that's what's called penetrance, and the penetrance of that disease is 1. And once you get it, you will not survive longer than a year. And you will be lucky to survive 6 months. Now, you could have an H46R mutation, which affects the metal binding, and survive 17 years, and maybe not get the disease, right? And then to put some neat stuff in the ointment to not make it sound so simple, somehow Scandinavian people all walk around, 5% of them, with a mutation that would kill an American with unit probability. So there's genetic modification on top of this to make it interesting. But, I mean, biology is never quite as cut and dry, but that's the exception to the rule. So since only 2% of ALS is caused by mutations in SOD1, we ask, what about the rest? And we just take the silly hypothesis, like, well, why don't we, if 150 different modifications anywhere in the protein sequence cause a disease, why can't a post-translational modification? Why can't you modify it with superoxide or modify it with peroxide? That's its substrate and product, respectively, right? So those are the kind of things we're asking and using the mass spectrometer to help us with that. And Jared will talk about this, that you can stabilize these proteins, and that's a therapeutic approach. Okay, I'm going to skip this. So I try to do this in one breath. I have to take a drink first. Okay, so I took a biomedical ethics course as an undergrad, and I realized that once I had my own funding, I could experiment upon myself. And all I had to do was send to my IRB. I clearly, by definition, have informed consent. You cannot if you work for somebody, but I have informed consent, and I hereby notify you that I shall be taking my blood and this and that and the other thing. So that's what I did as a postdoc because I wanted to purify the protein from people. The antibodies were very expensive, $3 a microgram. I couldn't afford them, so I post-haste began drawing blood, purifying the superoxide dismutase from my blood, using that to inject rabbits, immunize the rabbits made the antibody, and then all of a sudden I had $1 million worth of antibody. So that's how that worked. Wasn't too bad. The real loser in this were the rabbits. But once we had all that antibody, we could take the antibody, put it on beads. By the way, if you want to do antibody purifications, I can really help you with that. It goes beyond the scope of this, but we do that routinely. So can Jared, and we'll put this we'll make you antibody beads that you can use 500 times. We can teach you how to do that instead of just wasting them, and so that's good. So from that, I then took the antibodies. I put the antibodies on beads. I had all kinds of stuff to, I would sometimes throw some antibody at my lab mates, hit them in the head, and they would have to chase it because if they wanted some, that's how I gave it to people. I was really angry at the time because they said it would never work. So I was like, well, it won't work. Then there's your antibody. A little liquid nitrogen, frozen. Oh. So yeah, it's okay as scientists to be super petty. Once we do that, erythrocytes, if I purified from my blood, clearly I had a lot of it purified from my blood, and I was bored other than the first experiment I did, which I'm going to spare you. No, I won't. I had put protease inhibitors, and so the first time I purified my protein, it was modified by 183 AEBSF, and I was like, I'm going to get ALS. I took them out, and it was okay. That's not me this time. Okay, so erythrocytes looks good. Now, if I purify them from a patient sample, these are just single charge states, plus 14s, isotopically resolved. From a patient sample, spinal cord, you'd say, oh my gosh, look at this modification there. Oh, and look at this. And Jared's going to talk about this. He figured this out. I figured this out. And I'll tell you what they are. So they're post-translational modifications, and they're oh, here. Okay, well, I'll just tell you then, because I forgot I put this little segue. They're oxygen modifications of one tryptophan and one cysteine. And they're not what happens in vitro if you take your protein and you throw peroxide and superoxide. There's something else. The protein must be in a complex in vivo that's protecting certain residues that these were unexpected results. I'll show you in a few slides that I went in, not just got these results, because that's just, who cares what's modified in a person? We made mutations in those to prevent those modifications. Painstakingly injected thousands of neurons, unidentified. We didn't know what we were injecting, so everything was, nobody knew the identity of what they were injecting, and showed that once you stop this modification from happening, survival increased, so things that would kill cells stopped completely. So not only they exist in vivo, but they are toxic. That's so important to kind of, bridge that gap. And so, here's the other thing about mass spec. It can solve structures. No, it can't. But it complements other structural methods, okay? It really does. But if you're going to do that, that requires labels, okay? So you can do chemicals, so Jared will talk about that. You can do protons, which we've already talked about. The proton labeling happens implicitly during the mass spec experiment, correct? During electrospray in particular. You can put in oxygen. You can modify them with, you guys actually, does anybody do crystal structures? And anybody else use a synchrotron? See, you're already doing footprinting because you're making hydroxyl radicals and what other nasty things, and they're modifying your protein. Believe me, I've seen them when they come back. But you know that too because they're reducing your metals as well, and where are those electrons coming from? So some of you are doing this without knowing. But it's actually not, I'm not trying to be funny. Maybe you could take advantage of that. Right? Now, most people do footprinting in solution with an excimer laser or a short laser. Okay, or you can use deuterium. So here we've done that. So you have to cover, these are digests. So you do trypsin or pepsin digest of the protein, find out what each of these pieces are. And once you've done that, then you can label the entire protein with deuterium. And you can ask, how fast does this peptide as opposed to this peptide as opposed to this one exchange with solution? And you can make these exchange curves. And from that, you can infer that this is more dynamic, more unfolded, more solvent exposed, right? And you really infer, as people say, structure and dynamics, because you can't, from this, you can't decouple those. Okay? So we've published that. And what it looks like is this is that heat map onto a normal protein, wild-type SOD1, and then one that causes ALS. Now, here are the metals. Right there. It's a homodimer. And here's the area that's perturbed in this mutation. Well, it's a variant, actually, but you know what I mean. And we did this for all of these different ALS-causing variants. And lo and behold, X is where the actual mutation is, and every single one of them had a problem in the electrostatic loop right there by the metals. Oh, kind of cool. Just what they did. And clearly that's a great example of allosterism, right? Something's going on. I still can't tell you what's going on, but you mutate this thing way over here, and it perturbs the structure there. And there's also, these are all active. These are not. These do not bind the metals. So the ones from here down don't bind metals. So if you don't bind metals, you have an additional perturbation. So there was a common signature and then also an additional perturbation for things that bind metals. And that's been published. I'm going to spare you these molecular epidemiology studies, but this is the actual, in primary sequence, the two things that are oxidatively modified, this cysteine and this tryptophan. Again, I don't want to belabor this, but this is a mass spectrum. If you take that protein and you digest it, that's what you're going to see. You can zoom in on a single peptide and its oxidative modifications. You can isolate them, like I said, a mass spectrometer can do, purify them, break them into their pieces, and from the pieces you can find exactly where they're modified. Okay? Now this, if you try to do with metals, it won't work. They will have fallen off before they even ionize. Okay. So that's what it looks like on the crystal structure. There's that tryptophan. We can ask, by doing global hydrogen deuterium exchange, the intact protein is now in solution, okay? So here's at plus 10. Here's the entire spectrum. This is a metalloprotein. We're doing mass spec on a metalloprotein. So it has a very tight charge state distribution centered at this plus 8. If we put this in deuterium, we found out right away that we have 3 different conformations. You can, without doing any further analysis, you know that you have 3 different slow exchanging conformations, and that the thing that affects the conformation more than anything else is this modification by oxygen. So you know that already. Now keep these in mind. We're talking about, look, I've done the thing that everybody does where you fake the axes, right? So that's 78 to 123. In a bit, I'll show you what happens when you take the metals out. Here you go. If you take the metylated protein and do that global exchange, it looks like this. And if you take out the metals, it's really unfolding. Metylated wild type, apo wild type, and if you have something that causes ALS, it unfolds even more. Now, we've been developing, and other groups have been developing ways to not have to digest the protein, but within the mass spectrometer, sprinkle it with thermionic electrons and do different tricks to dissociate in the gas phase and find out where the deuterium are. And here's an unpublished, still hot result, but almost residue by residue, we can see where the deuterium are adding now and where the structure is perturbed. So in this case, ApoSOD1 is a mess, and everywhere, except for these 2. All right. What I do want to tell you is if you do break a protein in the gas phase, we can't learn today how to do this, but I'll tell you good news. In the last couple of years, we and another group in we, because they did it first, introduced database search engines that can do this for you. So this is what the actual dissociation of a protein looks like. This is actually hard to interpret. We don't have to. We can just plug it in with this mascot TD, which is published in Karabachek in 2009, and this is ours, and it will tell you what your protein is, just like a proteomic search, okay? And if you can maintain a metal, you can even program that in as a variable modification. So that's what you do if you want to look at the dissociation behavior, and it looks like this. Hey, this is your protein. It even tells you which mutant it is. We put in 200 mutants, and it'll even tell you which sequence variant. This is meaningful to you if you're studying something that's real from a mammal. Do you really know which isoform of your gene? Your gene may have 1 isoform. It may have 50 isoforms. Which one is the one that interests you? If you purify it in vivo, you should dissociate it and find out which mRNA gave rise to your protein. Right? The dogma used to be DNA makes RNA makes protein, and now it is DNA makes lots and lots of RNA, which makes even more protein in terms of the total numbers. Okay. And when we're done, we can use the mass spectrometer to say, here's our oxidized stuff on a cysteine. We can purify that, give it to a group that knows how to do toxicity, and they'll show us that it slows axonal transport. So it's actually, that's another cause of it being toxic. Here's our toxicity. I'm going to spare you that, just to say that here's what something, how fast cells die. with an ALS mutation, without, and then there's our rescue where we stop that tryptophan from being oxidized. People did the same thing with the cysteine. It's all straightforward. Now, this you have to keep in mind. Don't digest your protein, but if you do, know that a bunch of stuff scrambles and changes locations. If it's any sulfur modification, I'm showing you cysteine, but any S-thiolation. And lots of cool S-thiolation, right? Well, I'll admit. It's going to change residues as soon as you digest the protein. And here we did the experiment. So Jared had, flash forward, had actually solved the crystal structure. We knew where the cysteine was. It was only on one residue. We knew from top down, intact protein, it was only on one residue. But as soon as you digest it, it went to all of them. So if you weren't careful, you'd publish something that was entirely wrong and say, wow, my protein is cystinylated everywhere, modified everywhere. So keep that in mind when you digest. Also, I won't say anything more, but maintaining anaerobicity does the same thing. Can make or break modifications. Okay, so that's good. So that was the HD exchange. Cross-linking, Jared will talk about. And here we go. So I'm right on time, half an hour. And since I'm actually a minute early, here comes Jared's stuff. Do you want any questions at this point? It's a good time to ask. Oh, okay. So now I'll introduce Jared Auclair. He is being promoted now to research professor and also in the job search, just saying, and for faculty, and was hired to do stuff we couldn't do in the lab.
Great, thanks, Jeff. So I just want to give you a flavor for some of the things that I do with mass spec. And in particular, I want to focus sort of on some structural characterization of the proteins and protein-protein interactions that I'm able to look at. And I should say that I'm a joint postdoc with Greg Petsko and Dagmar Ringa at Brandeis also, so I dabble in crystallography. So the first thing I want to do is give you two examples of cross-linking mass spectrometry I've done. One, sort of a proteomics approach, sort of a low resolution, if you will, so I can try to convince you that we can do this with other instruments besides the FTMS that we have at Northeastern. And then I'll give you an example of intact mass, the intact mass approach that relates to SOD that we've been talking about. But I'm going to start with the proteomics approach, which goes back to, I don't know, like 10 years now for me, when I was a grad student at UMass with Celia Schiffer, who sort of let me dabble into my own projects as a first-year student, which was bad maybe back then and good now because I learned a lot more. So this is just a general cross-linking approach, right? So you have an isolated protein complex. It can be different proteins interacting with each other or, oligomers or whatever. And you add a cross-linker, and then you get a cross-linked protein. You can see some cross-links don't attach to other proteins. They just dangle off there. You have some that are, within one monomer, so those are intramolecular cross-links. And you have some that span between two different proteins, which are intermolecular cross-links. So then after you do the cross-linking experiment, one thing you can do is you can digest and do LCMS and look at your spectra. But what I'll focus on is this side, which is an in-gel digest, which of course is where we lost our metals. And at the time I didn't know, but the protein that I looked at in the Schiffer lab was an HIV protein, which binds zinc. So the first thing I did is I ran a uncross-linked sample on an SDS page gel, and then I cross-linked it and ran a cross-linked sample on SDS page gel. I cut out a monomer band, and then you get your spectra, and that's your peptide coverage. And then you do the same thing for the monomer band that's cross-linked, and then your different oligomeric states. And then in that monomer that's cross-linked, if you have new peaks, those are peaks corresponding typically to an intramolecular cross-link. And then new peaks again, which are denoted by this pound, are intermolecular cross-links. So this sort of experiment I did in triplicate and make sure all the data was accurate. And then you can funnel those in and use them as constraints in molecular modeling, or if you have a homology model, you can use them to validate that. There's also some bioinformatics software out there that will sort of build you a structure. I haven't placed much faith in that yet. I think that's someplace that can still be advanced greatly. But as Jeff mentioned, some of the advantages are that we can have information about distinct distance constraints, right? If you use different cross-linkers at different lengths, then you can know how far apart your residues are. You're not limited by the mass of the complex. It can be huge because we digested it. The really important part for me was the nanomoles of protein needed. Right, my goal as a enthusiastic first-year grad student was to take this protein that had never been purified or expressed and crystallize it, and then get my Nature paper and graduate in 3 years. But I can tell you, almost 7 years later, that didn't work out. But I did learn mass spec, which is not what I intended to do as a grad student, which brought me down the path that I followed, which I'm grateful for. So the other thing is cross-linking happens in solution, so your molecule can be dynamic. Some disadvantages, right, is you're increasing the sample complexity. Basically, you're looking for a needle in a haystack, which I'm really good at now, if anybody is interested. Some solutions, right, you can use isotopically labeled cross-linkers. You can use heavy water, which I'll mention briefly. And like I already said, there's still bioinformatic tools that I think need to be developed. So here's that gel, which mimics the previous slide. Here's the monomer that's not cross-linked. Here's a cross-linked sample. I excised the monomer bands and the dimer bands, and I did an in-gel digest with trypsin, and then I did just a simple MALDI experiment. When I did the in-gel digest with trypsin, I used heavy water, so instead of 16O, I used 18O. When the trypsin cleaves the protein, it's adding a couple of waters per peptide, so for a non-cross-linked peptide, you're going to have a mass shift of 4 Daltons, and for a cross-linked peptide, you'll have a mass shift of 8 Daltons. So here's just a regular peptide in the protein, which has that 4 Dalton mass shift, and here's another that has that 8 Dalton mass shift. And the cross-linking sort of complexity of the cross-linking can make the data hard to interpret sometimes. So then briefly, if you map those cross-links onto a linear structure of your protein, I have all the cross-linked intramolecular cross-links. Here are the N-terminus. Here's my sequence coverage. Here I'm missing a whole bunch of sequence because the sequence there is RKKR, so you can imagine the trypsin cleaving there a bunch of times. And then when I looked at the monomer versus, say, a trimer, it had the same intramolecular cross-links, but then I have a whole bunch of new cross-links. And you can see that most of those new cross-links are between the N and the C-terminus, with a whole bunch in the C-terminus. And then we later went on to show through some other experiments that this protein undergoes a disorder to order transition upon oligomerization. So that's why we're getting all these new cross-links in the oligomeric states, but not in the monomer. So now I'll go back to SOD and tell you about the intact mass approach using a high resolution that we're looking at, or we looked at. So the idea was that SOD1 has these two cysteines here, cysteine 111, and we were going to take advantage of those and try to cross-link the SOD1 dimer. So one of the hypotheses is that the SOD1 dimer dissociates into monomers, and then those monomers cause ALS as opposed to large protofibrillar aggregates. So if we thought maybe if we can cross-link that, we can in fact stabilize the protein and then in the long run prevent disease. So this is sort of a foray into pharmacological chaperones. And to do that, we use malamide chemistry. Here's one that we use DTME and BMOE, just so that everybody's clear. I don't intend to throw these into anybody. They're really toxic, so we'll avoid that. But hopefully we can use that to sort of build out the strategy, which is what's been happening in the lab since this paper was published also. So really quickly, here's the DTME structure again. You can imagine that if you cross-link SOD1, so here's the cysteine residues zoomed in, you can get this cross-linked cysteine 111 to the beta cysteine 111 through this malamide chemistry. The other thing that was interesting that we noticed is that thiodisulfide exchange can happen here. So this disulfide bond exchanges with the free sulfhydryl group on the cysteine, and then you get this. This is a model of what half of the DTME bound to the SOD looks like, which we also confirmed by mass spec. Excuse me. So here's the intact mass spectra of the cross-linked SOD1. Again, here's the pretty picture. So what I'm showing you on the left is an extracted ion chromatogram. So basically, I'm looking at 1 m/z eluted over time from an HPLC run, keeping in mind, again, right, that all my HPLC buffers have formic acid, so there's no metals present. This is for the ALS-causing variant G93A. For the non-cross-linked sample, if I look at m/z's for monomers, you can see that I have this nice peak here. So this is 1058. And then in blue is what would be a dimer m/z, and there's none. And then in the cross-link form, I have the amorphically related to the dimer, but none to the monomer. And these are just what the spectra look like. Here's a charge state distribution of the non-cross-linked and the charge state distribution of the cross-linked form. And here's the isotopic resolution of those peaks that are highlighted. So then I use those computer programs in my own math to calculate the molecular mass of G93A. So it's 15850, which corresponds to the theoretical mass nicely. And then in the cross-linked form, if you subtract the molecular weight of the cross-linker and then divide it by 2, because it's a dimer, you get back the molecular weight of the monomer. So in fact, we're looking at a cross-linked dimer with one cross-linker per subunit. So then, like Jeff said, we're interested in studying disease, and the mass spec is really cool, but so that's great. I stabilized it, but did it do anything to the protein? So one experiment that we're quite fond of is using differential scanning florimetry to look at the stability of the protein. So basically what you're doing is you're taking your protein, you're incubating it with a fluorescent dye, in this case Cipro orange, and then you're heating it up over time. And you can imagine as you're heating it up over time, the protein sort of denatures and that dye binds to hydrophobic regions. So this is a example of one ALS causing variant, G85R. It's metal deficient. In black, you're looking at the melting temperature of the protein uncross-linked, which is about 40 degrees. And then if you add increasing amounts of cross-linker, you get a shift in stability by about 20 degrees, which is, I still think, one of the biggest shifts in protein stability that's been reported to date. The same thing down here with the SOD. Here's the black trace, which is SOD by itself. Then if you add exogenous copper and zinc in this dotted line, just by doing that, you've increased the melting temperature by a little less than 40 degrees. And then if you add exogenous copper and zinc in cross-linker instead of a 20 shift, you get about a 40-degree shift in stability. So then we also were interested in looking at the activity. So you can see that, as Jeff said, most of the variants for ALS are active. So here's G93A is active across the board, almost as active as wild type. But G85R is not active. So here's the G85R by itself. So in this activity assay, it's a native gel. You run the protein and you stain it in the presence of nitroblue tetrazolium and riboflavin. Creates oxygen-free radicals if the protein's active. Again, it's a native gel. It leaves a white mark where the protein was. As you can imagine, the protein's traveling down the gel, so that's why it tends to streak occasionally. So if you add copper and zinc to the G85R, the activity is increased. If you cross-link it, the activity is increased, sort of on a similar level. If you add TSEP, the activity is lost. And then if you cross-link it and you add copper and zinc, the activity is increased to almost wild-type levels. When you break that cross-link, so this DTME cross-linker has a disulfide bond in the center. If you reduce that disulfide bond, you lose activity again in the cross-link form. So we were able to show that we had the one cross-link per dimer. It affected stability, increased stability, and it also affected the activity of metal-deficient variants. So there's some things that you can do beyond cross-link that I like to think about, because cross-linking, like I said, can be hard. You can get structural information from mass spec doing things like pull-downs, where you just, the typical experiment everybody does, you pull down a bunch of proteins, you digest them, and you identify them. You can use stable isotope amino acids in cell culture or SILAC that can give you some quantitative information. You can do non-covalent interactions or soft ionization. I only mentioned this briefly because this is how I was sort of attracted to the lab. Again, 6 years ago, I guess that's my number. 6 years grad student, 6 years postdoc. So then this is just one charge state for SOD that's unmodified by anything. If I add a compound and bind them, I can see that shift in mass here for this new peak. If you look for the compound bound, and the mass difference is that of the compound. So this is using soft ionization where everything stays intact in the mass spectrometer. Some of the voltages are lower, and we typically use ammonium acetate as a buffer. And the other things that you can do are ion mobility, hydrogen deuterium exchange, which I've talked about, and some footprinting. So then I just want to leave you with some thoughts about some complementarity between mass spectrometry and protein crystallography. I think that, especially nowadays, it's sort of bad to be a one-trick pony, so that's why, again, I took this postdoc as a sort of a mass spec crystallographer sort of thing, so I could sort of try to expand my horizons. So I'm going to give you some complementarity between mass spectrometry and protein crystallization, where we looked at it in vivo and in vitro post-translational modification of SOD1. So as Jeff already mentioned, here's some information about post-translational modifications. You can imagine one gene doesn't produce only one protein, but you're producing a protein that can be phosphorylated, cystinylated, acetylated, et cetera, et cetera. Typical proteomic studies remove some PTM, such as S-thiolation, due to alkylation and reduction. So here's sort of a theme. A lot of the mass spec experiments that are done, too, sort of remove the metals. So we're going to do an intact mass analysis because it doesn't typically use alkylation and reduction, and we can look at this S-thiolation or cysteinylation. And the intact mass allows us to determine how many PTMs are on the protein also. So I'm going to rewrite history a little bit in terms of the story. So here's the mass spectrum of SOD1 in the molecular domain. So I took those antibody beads that we created, and if anybody's curious, we still do bleed Jeff in the lab, just not daily anymore. So it's all good. So I took the antibody he produced in rabbits, and I've also noticed here at Penn State there's a ton of rabbits, so you probably wouldn't be sad for your rabbits. Everywhere I walk, I'm accosted by a rabbit. So anyway, so we have those antibody beads. I homogenize the human brain. I purify the SOD1, and then I looked at it in the Fourier transform ion cyclotron resonance mass spectrometer. Here's the SOD1 peak here at 15,844 is the average mass. And then you can see that it's oxidized, again, from human brain with 1 or 2 oxygens and some more here. And then we noticed this modification here, which was 119 Daltons away from the molecular mass of the SOD1 unmodified. And then we went on to show that that, in fact, was cystinylated. And so you can see here I recapitulated this sort of profile using SOD that I purified from yeast and oxidized, and then with cysteine added. This blue bar is showing a protection, which I'll talk about again in a second. Jeff flashed through the data, but we did show that this was actually cysteinylation by a top-down approach and crystallography. So here's my crystal structure of SOD. The one that's all rainbow is wild-type SOD, and the one that's magenta is the modified form. And then just zoomed in, here's the cysteine 111 region. Just zoomed in, you can see the electron density for the added cysteine at cysteine 111, so cis-cis. So this is the beta monomer and the alpha monomer, and here it is just zoomed in closer. Here's the cysteinylation. So then in vitro experiment, again, I showed that, so again, SOD purified from a yeast. Here's SOD1. Incubating it with cysteine. Here's the cystinylated form, 119 Daltons away. And then if I do the same experiment and add the substrate, the product, hydrogen peroxide, over time, you can see that I'm getting 2 or 3 oxygens added, and then after 4 hours, there's no more unmodified SOD, but it's all oxygenated. The interesting thing here, right, is that the protein that we cystinylated, again, is not being oxidized. So if oxidation is bad for disease, and we're trying to look for post-translational modifications involved in disease, typically we look for ones that might cause it, but this one might, in fact, be protective. And again, the same thermofluor assay shows that the melting temperature of wild type in the 80s, the oxidized form is destabilized by a good 40 degrees, and then the cross-linked or the cystinylated form increases the stability of the SOD protein. And then the crystal structure also showed that if you look at the subunit that's not modified, the dimer interface, they pretty much correspond nicely. The subunit that's modified, there's a shift in the beta of the loop 6, which is where that cysteine is. And if you look, scan the whole molecule, you see another shift at the electrostatic loop near the metal binding region. So then I just want to end with this one last thought about some S-dilation and post-translational modifications. And we were curious if the cysteinylation might be an artifact of the way we were purifying the protein due to molecular oxygen and cysteine hanging around. So here's SOD purified aerobically, the way we've always done it from a human brain. You can see that I have cysteinylation of the SOD1. Now we just took the purification of the human brain and did it entirely in an anaerobic chamber. And you can see there was a reduction in the amount of cysteinylation we observed, about twofold. And then again, we were curious if some free cysteine might be binding. So we incubated the SOD with alkylating agents, iodocetamide, iacetic acid, and cysteine scavenger, MMTS, to see if it would soak up any free cysteine and prevent cysteinylation further, again in an anaerobic chamber. And you can see the amount of cysteinylation still there, but reduced by about 50-fold. So just keep that in mind when you're looking at especially S-dilation protein post-translational modifications, that some of the molecular oxygen and other things that are happening might affect your PTM analysis. And I'll just end with this thought. Even as a old and crotchety grad student, maybe I'm not as old and crotchety as some faculty, but postdoc, right, it's always still good to listen to your mentor, because this was Jeff's experiment and his idea. I can remember thinking, it was a while ago, like, oh, really, do I want to do that in an anaerobic chamber? I don't even know what that is. Because again, I'm a protein biochemist by training, but I decided that, okay, well, we should do this anyway. I learned some new things, and we did it, and good thing we did. All right. So.
We'll end too with saying that what Jared saw with cysteinylation in this case, when I studied, we did a lot with the ISE proteins, ISCU, ISCA, and had a bunch of metalloproteins, and every single one of those